Dear audience, it's my privilege today to introduce our speaker, Claudia Langenberg. And we welcome her to presenting her talk on From Molecules to Medical Records, Insights from Large-Scale Multi-Omic Studies. Claudia studied medicine in Münster, and after an initial internship in internal medicine in the University Medical Center of Lübeck, she moved to the United Kingdom back in 2020, where she started an amazing career in the field of clinical epidemiology and public health. A year ago, she moved to our Berlin Institute of Health and the Charité to establish herself as full professor of computational medicine in our Center of Digital Health. Before, as an MRC investigator, she worked in several distinguished institutions in Cambridge recently, and before in London and San Diego, and served as group leader of the MRC epidemiology unit as a member of the University of Cambridge. Her CV represents a truly superb record of grant management achievements and publications. There are over 300 peer-reviewed papers listed. Her H-index will soon climb over 100, and she has far more than 40,000 citations already collected. There are several highly cited papers in famous journals such as from the Nature series, Lancet or JAMA. And also worth noting is a remarkable royal flush of additional outstanding publications over the past few months. So what is Claudia working on? What makes her so successful? Her topics include the identification of the genetic regulation of circulating metabolites and proteins, and the use of these insights to understand the causes of human diseases and providing insights into their management, including frequent important diseases. Prominent topics of her work include the connection of adipose storage to insulin resistance and the cardiometabolic risk, or the genetics of diabetes-related traits, where she made several important discoveries on genes that play a major role in type 2 diabetes. In her repertoire, you will also find then Mendelian Randomisation studies and studies addressing gene lifespan interaction, and recently expanding to large-scale multi-omic studies with detailed pathway coverage. No surprise that Claudia also serves on scientific advisory boards and committees of several highly important research institutions and activities. Furthermore, her work has already a tangible impact on health policy. Among others, she served as chief editor of England's Chief Medical Officer report on Generation Genome, published a few years ago. And this includes also discussions relevant in the ethical, legal, and social framework of genomic medicine. No surprise that she received a number of international invited talks, including keynote lectures. And so we can simply summarize that Claudia is an internationally highly visible clinician and medical scientist with leading role in the discovery of genomic regulation of metabolic control. Claudia, this was still an incomplete description of your achievements. We're simply proud to have you on board, and now the stage is yours. And finally, I would like to also extend my gratitude to Uli Dirnagel, who will later on moderate the discussion after your talk. In the meantime, you've read the policy for the talk here on the slide, and now Claudia can start demonstrating her work.
So again, thank you so much, Christopher, for the kind invitation and the introduction. So before I start, I just want to consider what the heck are these omics that everyone is talking about. So if you consider this schematic flow of biological information, you know, it's true to say that each of these components that I show here has been measured and in fact studied for many decades. But what really is different now is that technological advances have enabled us, or have enabled our ambition to really simultaneously assess the near totality of each of these. And that's what I refer to as omics. And not only that, this can now be done at population scale. And I don't just mean hundreds or thousands, but hundreds of thousands of individuals. And if you consider this for DNA, we've gone from variant to genome-wide genotyping, whole exome sequencing, whole genome sequencing in huge numbers. So the same, and just to end with the phenomic side, this is also now true for information that's embedded in electronic healthcare records. I want to take another step back and look at the evolution of genome-wide association studies. So here on the left, we see the very strong correlation between the number of associations that are found in such studies and the number of participants that were included over time. So here, 2007 is when I entered the field, and you can see this here, between 13 and 14 is when I was. So of course, that could just be coincidence. And on the right, you see the increase in the number of journals that publish these studies and diseases that are included. Sorry, could everybody please be on mute? Thanks very much. So for me, this was the beginning. This was my first GWAS, this was back in 2009. And things were very simple then. Sorry, there's still some people not on mute. Sorry. So things were very simple. Generally, there were very many more authors than insights. GWAS is a very, very simple tool. It's simply scaling the basic method to systematically test associations for all genetic variation that we can measure and really accept the penalty in terms of multiple testing. It is simple, but it's powerful. And the truth stood out. So this is a study where we investigated genetic determinants associated with fasting glucose, which is generally tightly regulated in individuals without diabetes. And we identified variants linked to 3 biologically plausible candidate genes. And here you see one of those, the melatonin receptor 1B. At the top is like a regional association plot. You can see the lead variant, a common lead variant, and the signal here within the gene. We found independently evidence that this was through reduced beta cell function, and at the bottom you see the association of the lead variant with the risk of diabetes in 10 independent studies. So as I used to say when I first presented this, if you look at the p-value of less than 10 to the minus 50, this is likely to be a true positive finding. And this really held up for many of the discoveries that we have made. So after decades of kind of hypothesis-driven approaches with very modest success and replication rate, this for me personally was an eye-opener that got me hooked. And since systematic and scale have become my favorite and probably most overused words. But where are we kind of more than 12 years later now? So despite 120,000 genome-wide associations that have been reported in the GWAS catalog, the mechanisms underlying the vast majority of all of these reported discoveries still remain unknown. And for common variants, we do not know the causal driver or variant, the causal gene, or the mechanism underlying this. And this is one of the largest obstacles to translating these findings into clinical insights. And this is why a lot of our work integrates these intermediate layers, the transcriptome, proteome, and metabolome, to really help to translate these genomic findings into clinical action or actionable insights. Now, we study very large populations, and that, of course, influences our choice of matrix in terms of the accessibility of it and the technology, i.e. the speed, if we want to measure thousands of individuals. So our focus on all of this talk is pretty much focused on blood-based omics, and I know that, of course, many people here study this in other tissues and cells, but we use scalable techniques that can be done in large populations, and they, of course, differ in terms of their coverage, their content, and their specificity, as opposed to focusing on a single protein or metabolite of interest. So I'm going to start for the talk at the distal end and consider the association of the metabolome on the phenome or disease endpoints. So as you know, and we're incredibly lucky here at BIH and Charité to have experts in the core, leading the core facilities related to various omics technologies, metabolome, proteome, sequencing. So it's a huge privilege to be in this environment. So for the metabolome, I don't need to explain many of you this, but it refers to the entirety of small molecules and biospecimen, and here we consider blood, and includes many of the standard clinical markers such as glucose and cortisol that we all know about. So this is a study where we looked at metabolome-wide disease associations, and it's embedded in the EPIC Norfolk cohort, where we included over 11,000 participants that had blood stored in liquid nitrogen and did untargeted metabolomics using a company called Metabolome that assesses up to 1,000 or even over 1,000 metabolites, but including here at the bottom in pink, a large group of unknowns, which means their chemical identity is actually not really been specifically identified. So on the top right, what you see are the 20s is the incidence, so new onset of 27 diseases that occurred after baseline that we assessed using record linkage based on hospitalization, so the more severe end of the spectrum, including over 200,000 person years of follow-up. And I just want to highlight one of the huge advantages in my view as an epidemiologist. So the near complete follow-up when endpoints are based on electronic health record linkage is a huge advantage in minimizing some of the biases and due to selection in many of these kind of so-called historical cohorts. So the plot at the top right is a sad plot, as I would say. These are these conditions, and each of the lines is an event that occurs, and at the bottom you see mortality, where of course in a middle-aged to older population after 25 years, many of the people have died. And at the bottom, you see all of the associations that we've seen, just here broadly categorized, just to show you how many associations you can detect if you scale and adequately power this study. Now, I want to just highlight that many studies that are published focus on a given metabolite and a specific disease. And I hope that when you look at this picture, you understand why I would say that that's not necessarily a particularly useful approach, because you may infer specificity that's actually in fact not the case. And this is what you see here, and I really love this plot. This is only for lipids. Around the circle, you see each of these incident diseases, and then you see in orange, bold orange, the larger these connections, the more metabolite associations are shared between diseases, and little gray part of each of these. So here, for example, is what is unique to that particular endpoint. So huge sharedness for lipids. You may say, well, that's pretty obvious. You consider cardiometabolic conditions, but in fact, we see this sharedness across all classes. In fact, 66% of all the hundreds of associations we saw were shared between diseases. Now, we know that metabolites are very influenced by our current state of health, genetic as well as other external factors, smoking, drugs, many other risk factors you can consider. And so this is what we look at here. This is these are considered over 50 risk factors where we had information at baseline, and look at the proportion of variants explained of metabolites that we had identified to be associated with what we refer to as non-communicable disease multimorbidity, i.e. the onset of 2 or more of the conditions that we considered. And what this really showed is that we identify what we call common antecedents of multimorbidity by saying that renal and kidney function, glycemic control, etc., underlie many of these metabolites we identified, and putatively they're linked to these diseases. So what's the message from this? It is that actually many of the risk factors that we may want to target to avoid multimorbidity are those that we already know about and manage in clinical practice. So that's the first one. The second one is, for me, that the lack of specificity of associations for a given metabolite or a single disease really shows the value of studying this, not only systematically, but at scale and bringing everything together. So I hope what this study also shows is that because of the strong influence of our current state of health on the metabolome, that if we're interested in inferring causal association, that adding genetic information can provide a tool for doing this, or kind of try to infer causality even within an observational setting. So here's an example. This is in a recent paper that kind of looks critically at this technique. It's useful to consider this. Many other studies, methodological studies, have been published. So if you consider a question, and often these questions arise from observational data that create a kind of statistical link. So the question is low cholesterol associated with an increased risk of cancer? Now we know if we study people with low cholesterol, there are many other factors that affect cholesterol levels that may also affect cancer risk. Very hard to draw causal inference. So the tool is then simply we look at the genetic susceptibility to high versus lower cholesterol level to then use this, apply it to studies that have studied cancer risk in genetic studies, totally independent studies most of the time, but adequately powered to look at, is this a causal association? And the advantage is that the genetic susceptibility as a tool means first we can avoid or minimize reverse causality. We know the variants, the cholesterol levels precede the cancer, not influenced by the onset of cancer. It's the first. The second one, the many factors that could otherwise confound the observational association are likely to be independent because of the mode of inheritance of these variants that we use of these other factors. And that's actually also something that's testable, whether our genetic instrument for low cholesterol is associated with other factors or how specific is it for this. So these are, of course, some assumptions that we can test. Just to illustrate this, one example, this is a select trial, so this is here a metabolite example. This again was inspired by observational associations of a link between selenium and prostate cancer risk. So it's a large randomized controlled trial. This is only the selenium versus placebo arm, but there were over 35,000 men included. It has cost over 100 million. And people were randomized in for this arm to selenium supplementation and versus placebo to see whether this could prevent the onset of prostate cancer. Overall, the trial was negative. And I think actually this estimate was after 8 years, but just to stretch, during no time of the follow-up up to the end was there a significant difference between the two groups. If anything, the cancer risk was increased or suggestively increased, but as I said, not significantly so. So now we use our tool and we know, and in this case we didn't discover these from scratch, but they're already reported in the literature. We use 11 variants that can identify in the population who's exposed to higher versus lower selenium levels, and we can use this as the tool in a less confounded manner to look at prostate cancer risk. And the result that we got, not just for prostate cancer, but also in this case for this collaboration, we looked at other adverse outcomes that had been reported, such as diabetes. The result is the same. And I think it's fair to say many of our talented students and postdoc, given that so much of these data are now openly available, could do this in an afternoon, and none of them is paid 100 million for this effort. So I don't want to say at all that this can replace trials, but what this can really crucially and rapidly do is to prioritize interventions to be pursued in trials. So it's a tool and it can be useful, but assumptions have to be met. So one of the things that we need to be in place is you have to have genetic instruments that you can use to infer causality for a given for given exposure. And in this case, we're still talking about metabolites. So we're lucky in the MRC Finland cohort. We had over 10,000 people where we measured targeted metabolomics using Biocrates. Biocrates P180 kit is, we assessed 174 metabolites. Everybody was genotyped. So first we set out to do a genetic analysis or discovery for each of these metabolites. Now, the simple trick again was, well, we could stick to our 10,000, but what if these metabolites, who we knew are also captured on other platforms? So this shows you the overlap between Biocrates, the untargeted metabolomics, the platform that I previously spoke about, or NMR, which you see here. So there had been studies had shared data in the public domain, and we had access to collaborator data for metabolites from these various platforms. So the question is, can you integrate genetic associations for metabolites measured using these three different ones? This leads to this kind of slightly awkward and unusual study design where you see at the top amino acids, which is kind of the lowest common denominator captured on all platforms, has massive sample size with over 85,000, down to biogenic amines at the bottom, which are specific to Biocrates. Of course, these are not all. This is collapsed just for presentational purposes. So what did we find? If you scale up and consider its different sample size for each of these, a lot. So 144 regions, almost 500 locus-metabolite associations. And just to show you here in this 3D Manhattan plot, for those not so familiar with this, here are the metabolites ordered by class, and here's the genomic position. And just to point out a few key things that we found. First of all, you see genomic regions here associated specifically with a given metabolite or metabolites within one class. But also you see here these kind of very busy lines with high peaks, that's the significance. These are very pleiotropic regions, so GCKR or fats associated with many metabolites across diverse classes, so not specific. Just to point out a few of the features of these kind of, we call them MQTLs, metabolite QTLs, these variants affecting metabolites. Here you see the association between the effect size, so this is standardized 1.5 standard deviations. Here's the minor allele frequency, so as you would probably expect, rare variants, low frequency rare variants have larger effects. This is colored by their functional, potential functional consequence, but you also see many common variants with very large effects. So a standard deviation difference is not insubstantial. Here, the horizontal line is just for reference, the effect size of the strongest common BMI-associated variants, FTO, that you may have heard about. So here, in this, consider this audience and all of us, even for all the people without existing disease, huge differences in metabolites between all of us. And the question really is, does this translate into a clinical entity? At the bottom, again, just very briefly, if you compare the variants that we identify, these MQTLs, to different sets of comparator SNPs, we see strong enrichment for exonic non-synonymous variants. And the same is true also for protein QTLs. So I only have time, I'm just going to check the time, I only have time for 2 examples. So the first example here is a missense variant in GLP2R, the glucagon-like peptide 2 receptor that we identified to be associated with citrulline levels. Now, GLP2R is expressed in the gut, is closely related to the glucagon receptor, and GLP1R, which is the target of insulin secretion, GLP1R agonists. So here's you see the Manhattan plot, and this is the signal. GLP1 and GIP are incretins, stimulate insulin secretion upon oral ingestion of glucose, and GLP2 is known to stimulate intestinal growth, and analogs are used to treat short bowel syndrome. Now, there was a study that came out, this is actually an RCT, so a randomized trial, which looked at the effect of tetraglutide, glucagon-like peptide 2 analog, and really provided strong evidence that citrulline can be seen as a biomarker of intestinal function and target engagement. Now, just as a bit of context, so we had previously identified a variant in high LD, or kind of in almost perfect correlation with the missense variant we identified for GLP2R, as to be associated with type 2 diabetes, one of our core interests. And if you consider here, this plot on the top right, this is when we zoom into the genomic region, so kind of the regional association for the various different entities we looked at. So here's the association for plasma citrulline, type 2 diabetes, BMI, and also GIP, the other incretin. And you see, and you can use a statistical technique that I'm going to come back to at the end called co-localization, where you really look, is this a shared signal in this region across these different traits? And the answer is yes. So this is the same signal driving this. And that begs the question, what about citrulline levels in type 2 diabetes? And for this here, you look in panel C, we, of course, as you see in the Manhattan plot, had several kind of loci identified to be associated with citrulline levels, but in fact, none of these, other than GLP2R, except for ABO, which I should say is a known very pleiotropic locus that we can ignore for now, none of the others is associated. And we interpret that to mean that this is the locus or gene-specific effect and not about citrulline levels in blood per se. Now, so in combination, we concluded from this that the carriers of the missense variant with higher citrulline levels, that this indicates greater GLP2R signaling, which in turn leads to chronically elevated GIP levels via greater enteroendocrine mass and GIP secreting K cells. And this in turn independently has been shown to be associated with reduced GIP receptor signaling in beta cells, which explains the association with reduced insulin secretion and increased risk of type 2 diabetes. Now, we were very lucky at the time, this was within the Institute of Metabolic Science in Cambridge, to collaborate with Fiona Gribble and Frank Reimann, really kind of, specialists on the physiology of incretins. And so because GLP2I is a G protein coupled receptor, the question is whether the association that we saw may be due to G protein dependent cyclic AMP signaling or independent beta-arrestin mediated signaling. And what we were able to show, or better what Fiona and Frank were able to show in their in vitro model, is that the mutant carrier had reduced recruitment of beta-arrestin upon GLP2 stimulation, and no association was seen for CAMP. So we therefore concluded that reduced recruitment of beta-arrestin or beta-arrestin mediated signaling is underlying the link between the variant and type 2 diabetes. Now, just very briefly, my second example for this. So staying with metabolite associations. So at the time of doing this study, another study emerged in the literature, which was a GWAS of a rare degenerative retinal disease called macular telangiectasia type 2. This was Melanie Barlow and a team, many Australian scientists, and they already speculated, because they only had few hits, few hundred patients in this study included. It's a rare disease, rare severe disease, that the loci they identified, at least 3, looked as if they were associated with glycine and serine metabolism. Now, because we had done the association studies, or genetic discovery for serine and glycine, we considered, so could this be useful to investigate this question in more depth? And kind of at a similar time, another study appeared, again based on rare disease patients where low serine bioavailability through levels of atypical deoxysphingolipids was linked very specifically via a few genes. So the question really stands, is this something restricted to these genes, or could we see this and a link between serine metabolism or serine glycine metabolism that kind of co-regulated and the risk of MECTEL type 2? And this is what we looked at here. In this plot, each of these dots represents a combination of all the genetic variants we identified for each of the 174 metabolites, and we relate this here on the y-axis to this is the log odds ratio for the risk of MECTEL. So you see that 2 really stand out, and no surprise, the top is serine and underneath is glycine. So very specific, considering we tested these 174. And now you drill into each of these dots, because of course many of the variants contribute to this kind of summary estimate, and you see this very strong dose relationship when you look at the individual variants. And if you again look at the y-axis, this is now the log odds ratio for MECTEL. It's a massive effect size, absolutely massive. And so because of this, in most epidemiological studies, effect sizes are modest or small and really not necessarily very useful for prediction. But because of this massive effect size, we considered, could this be useful? And again, in collaboration with Melanie and her team, looked at what the addition of our identified variants for the prediction of MECTEL could be, and this is what you see on the right. And what this, together with the other studies, has led to really is now that there are trials underway which investigate the effect of serine supplementation in individuals at high risk of MECTEL. And to conclude this part about the metabolomics, this was a targeted approach, and really in parallel we've conducted a study based on Metabolome, as I said at the beginning, what I started with at the beginning, where we look much more broadly at the genetic architecture of up to 1,000 metabolites, including the unknowns, in blood or in plasma. And what this enables us is, if you look here on the right, to really kind of map our genetic findings onto these metabolites and pathways. Again, this is work done in collaboration with this specific element with Metabolome. So watch that space. So to summarize the first part of my talk, I hope I've shown you that the increased scale and greater variant diversity has increased the number of identified variants for metabolites, that it is feasible to integrate genetic data on metabolites across different platforms. I didn't mention it, but there were very few examples where we observed heterogeneity of associations across these platforms. There were some, but very few. I've shown you specific MQTL characteristics and hopefully some examples of the potential clinical utility and improved understanding of disease mechanisms. I want to mention that in collaboration with Gabi Kastenmüller's team at the Helmholtz Center in Munich, they've helped us to design a web server where we share all of our results with the scientific community. So I'm going to change tack slightly and now move on to proteomic studies. And again, initially I want to focus on the right side, so the relation of the proteome to the phenome. So if we consider, again, we're staying with plasma, it's a matrix is relatively easily accessible, so we consider the plasma proteome. So of course, as we all know, proteins have very key roles in very diverse biological processes. They're changed or dysregulated in disease, and they're one of the most important class of drug targets. Now, one of the complications is that the concentrations span, a huge range, and hence, because of this dynamic range, there's no single technique that's currently able to provide reliable measurements for all proteins at the same time. And because we study large populations and we are reliant on technologies that we use to be scalable, these are the three that kind of at the moment, in our view, have been used by us and at the moment really stand out. So the first one is mass spectrometry, which is clearly considered the gold standard, which now also can achieve very high throughput, but really focuses on the abundant end and does lack some sensitivity for less abundant but biologically very active proteins, such as cytokines or some of the hormones. And then two of the other technologies or companies that we have worked with, you see in the middle is the proximity extension assay. It's like antibody-based, similar to kind of what we use, with clinical chemistry, is a company called Olink that provides in the range of up to recently 1,000 proteins and now extending up to 3,000 proteins. And finally, in panel C, you see the microarray technique for aptamers. It's a company called SomaLogic. They use short oligonucleotides, or so-called aptamers, which kind of match the 3D conformation of the protein, i.e. it's not so much necessarily the blood level of the protein that you can infer to, but more the affinity of the aptamer to bind to the 3D shape of the protein. So that's important. All right, so the huge advantage, just to highlight a few key features of this Finland study that I mentioned before, is that not only is it relatively large for this type of in-depth assessment, but people are, they're born between 1950 and 75, so I still call this young to middle age. And so as a consequence of that, many of the people are disease-free and treatment naive. And that's, of course, an advantage if you want to minimize the influence of existing disease on protein levels. So most of these were genotyped, and we've done, used the aptamer technology or SomaLogic on all of these individuals. And we've designed a sub-study with 500 individuals where we've measured all of the Olink proteins that were available at the time. And also, in a fantastic collaboration with Markus Reiser, initially at the CRIC and now here at Charité, do mass spec proteomics in the same people. Actually, the 500 people we did with 4 different samples during the OGTT and at a later time point, using scanning swath that Markus has really kind of brought to a high throughput that's now suitable for such huge, not just this size, but much bigger studies. And of course, the cost of that is substantially lower than the other technologies. So the target for this collaboration is not to remain at the 500, but scale this up to everybody. And it's currently kind of going on. So only as a quick reminder, so I'm going to switch on to SomaLogic now, and that's what the remainder of my talk is about. And just to consider what we're actually looking at, if we look at the SomaLogic, the 4,775 protein targets that we measure, so only a small proportion of these, just over 10%, of course, is actively secreted, such as coagulation factors or cytokines. But many of the others that we can measure are products of cell leakage or turnover or soluble fragments of membrane proteins. So it's, of course, it's what we measure in plasma is a mix of the tissues of origins and cells, and a huge influence also exists on the sampling procedures we have in place. And I don't just mean at the point of measurements, but at the point of blood collection and time to spin and time to storage. So it's just as a reminder. So what can this be used for? Or is this useful for? So this was a collaboration with SomaLogic, and really their USP is to develop insights based on plasma proteins that can predict diseases. So this was the first as a proof of principle study that they really led and focused on cardiometabolic conditions. So basically starting with nothing, not even clinical parameters, not even age and sex, but just using the proteins to see, can we assess, can we predict cardiovascular, cardiometabolic entities. So the answer was yes, this worked. It worked really well for most of the outcomes we considered. But what I would say is, and they've since, of course, expanded this, the initial insights and the commercialization was almost entirely based on our findings from the Finland study, but they've now moved on and included additional diseases they consider. But the point is, if you do this for clinical entities, diseases, risk factors, that we already measure really well in the clinic, there's little point in throwing so much money at this in order to gain a small additional improvement over and above what we can already do. So what I think is much more interesting is, of course, consider conditions that are currently poorly diagnosed, poorly detected. So can proteomics help in doing better for those? So one of my interests is focused on isolated post-challenge hyperglycemia. So what I mean by this is like glucose levels elevated 2 hours after an oral glucose challenge test. We know that this is strongly predictive of cardiometabolic diseases, and not only this, it's rarely measured because of the complexity and logistics and time, and also it is very common. So globally, it's estimated that 7.5% of adults are affected by this. I always have to think twice about this. It's a huge number globally. And importantly, unlike fasting glucose, which is relatively stable as people get older, and hence a diagnosis on the basis of fasting glucose doesn't change as much with age. But if you consider 2-hour glucose, it raises quite steeply. So if the definition was based on 2-hour glucose, which clearly is associated with complications of hyperglycemia, older people and specific ethnic groups are disproportionately disadvantaged. So we estimate that a large proportion of these has isolated post-challenge hyperglycemia, which is the condition when even if you measure fasting glucose, even if you measure HbA1c, these people are not detected. So the first time they present is when they start to have problems with their eyes or kidneys. So the question is it feasible to identify fasting proteomic signature to design a simple test that predicts this isolated IgT? Again, I return to the Finland study. This is Julia, very talented student, and so she divided these people up into those who have impaired glucose tolerance and those who have isolated impaired glucose tolerance. And she divided the Finland study up into one half that was used for feature selection, identification of proteins associated with these entities, and then a quarter each for parameter optimization and the evaluation of the model that she tested using Lasso. So if we look at the results, and again, I don't want to dwell on this for too long, but just to show you on the right, the results for the isolated IgT. And in fact, I should say this includes very few individuals in the Finland study, which excluded people with prevalent diabetes, but very few who had biochemical diabetes. So that exceeded the IgT value for 2-hour glucose. But so the first point is, even considering the best clinical model, so this includes what we call by Cambridge type 2 diabetes score. So these are parameters that we have in the health record that we could get to, and without calling the patient in, predict their risk of diabetes. To this, we added fasting plasma glucose and HbA1c, and even if we do the best we can, this is the AUC is only 0.75. proteins alone, 73 proteins alone achieve an AUC of 0.85, so a massive improvement, and only 3 proteins do much better than the best clinical model if they are added. So this seems doable. So this was, I don't know what to expect, but we were surprised by how well this worked. And just to kind of finish this part, one important feature. So if you consider the proteins that ranked highest in the feature selection for either of these outcomes, again, we used a little trick. If you look at panel B, what we consider here is not a kind of the measured association, but we derived genetic susceptibility scores for a variety of risk factors associated with diabetes. And what we observed was that the proteins that we detected were all bar one affected by genetic susceptibility to these risk factors. So they reflect our inborn risk to these metabolic disturbances. So this is what we identify with a totally different approach. And all bar one were associated in an independent study with the onset of incident diabetes. As I said, the study we derived this in was free from people with diabetes. And this, we've now confirmed this in the second independent study. So these proteins reflect our genetic susceptibility. And this brings me back to the last part of my talk, where where I come back to the fact that our technique and approach for trying to identify causal disease association is to go via the genome and use causal genetic evidence to infer this. So this seemingly easy or pretty, I would say, plot represents years of work and this huge complex undertaking. So I think it's still fair to say that this is probably the largest and most comprehensive in terms of proteomic coverage study of this kind. So where we did the genetic or defined the genetic architecture of each of the aptamers included on the SomaLogic version 4 panel. So what you see here on the x-axis is the position of the genetic variant, and on the y now here we have the position of the protein encoding gene. And what we show you for each dot are all of the aptamers where we have at least one variant significantly. In this case, this is much lower than just genome-wide significant, because of course on top of that we test 5,000 proteins that are detected. So if you consider this, the pink dots here are those where the position of the variant and the protein encoding gene converge, and that means these we refer to as cis-pQTLs, so i.e. within or in close proximity to the protein encoding gene, and the blue ones are what we consider trans associations in further away genes. And if you consider some of these lines that go up, here you see this is what I said previously for metabolites. These are extremely pleiotropic regions, so you see this includes the MHC region or ABO. And what you see on the right is simply what each of these dots represents, the association between a genetic variant and the aptamer, or the affinity of the aptamer to bind to the protein. So this is the summary plot of this endeavor. I just want to highlight, so everything I've said so far with the slide before kind of leads us to this. So we identified over 10,000 associations with these proteins, and of course in some ways we could stop there and go home for tea, as they say. So what do we do next? And really what is the exciting thing, and the thing that really opens up new avenues of investigation and for clinical translation, is the fact that we then integrate a genetic association across the whole of the human phenome that we either had access to, could get our hands on, or is available in the public domain. And so I come back to what I referred to earlier. If you look here on the bottom left, is now initially we consider all the kind of cis associations, so over 1,500, and we use this statistical tool co-localization to look at the local genetic architecture of the gene, protein, each disease triplet that has a very high posterior probability of being a shared genetic signal. And what we can infer from that is, if we find, for the examples that we found, is that the protein encoding gene via the protein is very, very likely to be the disease-causing gene. And we also considered continuous traits. And we integrate all of this evidence into what we call a Proteo-Genomic map of human health that includes over 1,800 connections. So from protein to different diseases, genetically anchored. So one gene, possibly one disease, possibly many diseases. So this is not yet published. So I only give you one example to just illustrate why we are excited about this. So this is the map here, and here we zoom in, and this is for a protein called fibrillin 3, which is encoded. So it's an extracellular matrix glycoprotein, which is encoded by a gene called FMP1. So what you see when you really look at this in detail, how this glycoprotein is associated with what we call convergence of a large number of soft or loosely defined soft or connective tissue disorders that kind of converge here around this protein encoded by this gene. And what this is based on, so you see here osteoarthritis, carpal tunnel operation, there was also syndrome, vaginal prolapse, extreme height, hernias, and so on. So what this is based on, just to remind you, is not loose connections based on a single SNP association, but here on the left, this co-localization where you really drill into, in the vicinity of this gene, is this the same signal across these many, I think in this example, it was over 37 conditions that are linked via this one gene and protein. And so the hypothesis here would be that the lower levels of the variant lead to an accumulation of intracellular protein concentrations and abnormal elastic fiber morphology. And this actually is in line with what we know about the FMP1 knockout mouse, who also shows this abnormal elastic fiber morphology and an increased risk of hernias, among other phenotypes. So with that, I want to summarize the second part of my talk, and I hope I've shown you the value of the plasma proteome for prediction, not just kind of common complex conditions, but also kind of conditions hard to diagnose or predict, such as isolated IgT, the value of increased breadth and scale if you're interested in defining the genetic architecture of these omics traits and proteins, what the integration with phenomic data can really achieve, illustrated by this genetically anchored disease map. So in terms of next stages, I just want to mention a few things while you are all here. So the first one really is a collaboration with Harry Hemingway and Spiros Danaxas, and hopefully people here at Charité to consider what we refer to as the neglected phenome. So the neglected phenome is we have very many very rare diseases, people focusing on the very common diseases, and in the middle is a huge number of representations that are seen here at Charité and throughout the world that people haven't really focused on because they're hard to get at. But now that we have electronic health record from primary and secondary care, we can use this, we can analyze it and integrate it into our map to find new genes causally associated with these diseases that people have really not looked at in detail. So that's the first thing. Secondly, excitingly, so we've applied this to the data that is available, different definitions of COVID-19 prognosis. I'm very grateful to my colleagues here at BIH and Charité. So Irina Lehmann, Christian Konrad, Søren Lucassen, and others to kind of really go from the epidemiological observations, the genetic associations, to the single cell sequencing and trying to add, biological insights to the observations we've made for certainly at least one new gene associated with COVID prognosis. And finally, something I would refer to as the 3C, which is the collaboration for clinical curation of the gene list that we have in our map. Now, the diseases we integrate, they span almost all clinical specialties. This goes far beyond what we can delve into. So to take this to the next stage, I hope that people like you, expert for specialties, certain conditions or genes and proteins, would have an enthusiasm to come together and go through this to define what the next step could be to really make additional insights. So again, the data already for some work is in the web server, so I encourage you to look if you're interested in specific proteins, and more will follow in due course. So I want to end with thanking the very many people who've enabled this work here, but also in the team in Cambridge that I have, and in fact all over the world. That includes many colleagues and also very good collaborators in industry. And finally, thanks to all of you for your patience and your interest. Thank you.